In this video we're going to be looking at gram staining using commercially available kits. There are a number of such kits available. Starting with a fresh clean microscope slide, add 50 microliters of water and then pick a colony from an overnight plate and mix it with the water. Make sure the colony is evenly dispersed. Alternatively, 50 microliters of a fresh overnight culture can be pipetted onto the slide. Next, the slide is dried by placing it on a 60-degree hot block. This will cause the media and the water to evaporate and eventually leave two dried areas of bacteria. The slide can then be transferred over to the sink where the staining tray is in place. First, crystal violet is put onto the slide and left for 60 seconds. This will stain the cells purple. Excess dye is poured off and the slide is washed, taking care not to wash directly onto the bacterial areas. Once the slide is washed, it is returned to the staining rack. And next, grams iodine is added to the slide. This is left for 1 minute. And after a minute, this again is rinsed off. The grams iodine forms an insoluble complex with the dye. So again, we rinse off excess stain and then return the slide to the rack. Next, we add a decolorizing solution. This will remove the dye from the gram-negative bacteria, leaving just the purple gram-positive bacteria behind. In the final stage, we add to the slide the safranin counterstain, and this will only be taken up by the bacterial cells that have lost the purple dye. Finally, we rinse the slide and the stain is complete. Next, we can view the slide under the microscopes and see the different types of staining. The gram-positive bacteria, which did not lose the crystal violet purple dye, will appear a purple colour under the slide, whereas the gram-negative bacteria that have lost the crystal violet iodine complex will appear pink.